Click for IB Biology. What we're looking at here are models of population growth. There's a blank version of this presentation, a PDF, down in the description. You can annotate that with your own notes. There are 10 questions with answers, and there's also an AI prompt that you can use. Copy and paste that into a chatbot, and that will give you individualized assessment of your own learning. So let's take a look at theoretical population growth. Ecologists will, when studying a species, will always attempt to create models of the population growth based on their data and the factors which are affecting that growth over time. We're going to begin our look at those models by considering the concept of exponential growth in which there are no limiting factors. If we just take a look at the factors to begin with, limiting factors on population growth can be divided into what are called the abiotic factors and the biotic factors. So perhaps at this stage, these are the non-biological and these are the biological factors, and you can see them listed underneath. And I'll return to this notion in a minute. But for our exponential growth, we have no such limitations at all. So the environment is placing no restriction on the reproduction and the population growth of our species. And perhaps this is observed within the laboratory, where we have optimal conditions for a species like this one here. This is actually a bacterial species like E. coli. And in the laboratory, we could begin with an initial animal, a bacteria rather, and it could go through a series of divisions as the population begins to expand and develop exponentially. And if that occurs with population on the y-axis and time on the x-axis, we would end up with this exponential upward-facing curve like this, which is indicative that none of the factors which are listed here are having any effect on the population. Indeed, the experimenters have removed those. Such conditions are highly unlikely to occur naturally under any conditions, and if they do, then it would be very briefly in the development of a population. The rate of development of such populations, again theoretically, the number increases are so radical that we would end up with numbers diverging and increasing on this exponential scale. And when ecologists encounter such numbers or something approximating to this, they prefer to plot instead the population on a logarithmic scale against time. And the purpose of this is it allows us to more readily incorporate a wide range of values from small numbers to very, very large numbers. By linearizing the exponential growth, we can formulate equations to describe them more easily. The data can be compared on different scales. That's another feature of logarithmic things. Percentage changes can be handled within this way as well. It de-emphasizes outlying population numbers, allowing the trends to be more easily determined. And by the same token, it enhances the sensitivity of the low values on a representation in which the researcher can determine the population size. Populations, however, rarely perform in such a manner, and they are in fact affected by what are called limiting factors. Here you can see I've split them into abiotic and biotic, and if we jump very quickly to the lower curve, we can say that most of the abiotic factors are density independent factors. So the size of the population plays no part in the impact of these particular features here, which you can see they're all physical aspects of the environment. The number of individuals in the population, however, does affect the acquisition of food, the levels of predation, the levels of disease, and finding reproductive partners. When we introduce those and we follow population growth, again beginning with an initial colonizing population here, allowing the population to expand into the environment, it's a theoretical idea, but with abiotic and biotic factors operating, then we don't get exponential growth. We get what's called sigmoidal growth with this sigmoidal curve here. And the difference between the two is called environmental resistance. So the difference, and it's a decreasing, factors are decreasing the population size, and if we go looking for what are the environmental resistant factors for a particular population, we would need to determine whether they were abiotic or biotic factors affecting our population. The sigmoidal curve has a number of descriptors that are associated with it. The early phase in which the limiting factors are not really restricting the population, or less so, is known as the exponential phase. Then what happens is the population rate increase, the rate of population growth begins to slow. This is called the transitional phase, so limiting factors are beginning to take effect. And then we reach the plateau, which is known as the stationary phase. And the plateau coincides with this point on the axis here, where the population size would be measured there for this particular environment, and this is called the carrying capacity of the habitat. So the population size of a species, once it reaches the stationary phase, is called the carrying capacity. And should any of the abiotic or biotic factors change, then that carrying capacity will change as well. And typically, when we observe such factors operating, abiotic factors will tend to abruptly affect the population independently of any population size anyway. So it doesn't matter what the population size is at any time. These density independent features such as natural disasters, aspects of climate, perhaps human impacts on them, and environmental changes, drastic environmental changes, can abruptly decrease populations irrespective of what phase of the sigmoidal curve that population has managed to achieve. On the other hand, and notice this axis here is different. This is population density, and this is the population growth rate. So as the population increases, density-dependent factors have a proportionately greater effect. And this is because these biotic factors are such factors such as predation. So if the number of prey is affected by a predator, the predator is the density-dependent factor, and the effectiveness of predation will very much depend on the number of prey which are available for successful feeding by the predator, and we call this a density-dependent effect. So they have two different types of effect: abrupt changes from abiotic, density independent, and more smoothed out transitional changes where we have biotic, density dependent changes in the population. Here's the link to the next video in our population community study. There's the link to subscribe to the channel and the playlist as always.